All right, it's running.
Yay.
Okay.
Oh my gosh, I believe you got this. This is a good question. Today is, what day is today? Today is
The second.
exactly, yeah. Okay, it's October 2nd, 2025. The time is 9:26 and this is the genetics laboratory.
We did the animal thing, and I would look at feather. Okay.
You guys, sure?
5 milliliters of blood suspended here, so
Oh, binding. Okay, so this is the other buffers are here. We have color coded them. For the NanoDrop, yeah. For the NanoDrop, you like the debuffer. Other than that, you won't really need the debuffer. But you have here, so PF for binding buffer, right? PF for binding buffer, and WB was wash buffer, and EB was a buffer. Okay, so it's actually, it should like it's yellow, blue, red. Is what you would choose. And here's the magnetic. Oh, you just try it. And you got one of the racks? Yes. Okay. Yes.
This thing right? No. The cooling.
Yeah, I just stole it. Oh, perfect.
He was standing there watching you. Did you
So you can find that time to 10. And 2 steps. But
okay. So combined temperature. So there it is.
90. And then we're gonna, are you ready? We're gonna walk them. They're still chill, but they can take a liquid. We only need to purify sample. I think we're doing all of them, even the negative control. But yeah, maybe we gotta do the same thing. Even though we have
Did you guys work together to make like one or like how?
Yeah. So, what's your name now?
Kayla.
Kayla, I'm Haley. Nice to meet you. Let's start with the... Okay, we want to end up... Do we have the binding buffer in the 90 set?
I didn't do a 10 because I wasn't sure. I didn't want to
We can always set this to 10. We need to have the 90. Yeah.
This is why they're good point. Thanks for pointing this out. If you're at the Qubit stage, then these are the tubes you want to use, please. They come actually in a clean in a sterile bag. So don't like no grabbing as you
I'll grab it. I can grab a bead. Okay.
I can check it.
Perfect.
Do you need any more of these?
Yes.
Yeah, I thought that was you.
You already have a waste heater on your
So this will be okay.
These should work.
And what do the bucket beads look like?
By the way, little hint here. I see several people already using the really small 0.5 millimeter tubes. I mean, this is, for example, how the primers come. If you want to have actual racks for that hold the small 0.5 millimeter tubes, be aware that the I think maybe you've already realized that the two small beam racks, these basically the one that Luis is using, you can use those as holders. Where are the other ones that we're using? Oh, you have the 40? Oh, we have plenty more in the back. Okay, no worries, I'll bring more.
When should I be ready?
Oh, do we have a waste Which is
And then do you want to add the silicone beads?
What?
Do you want to add the...
Yeah. Which one is what?
Okay, that's
I don't know what that
I think you can just use that one.
Okay, then.
After. When you do have the family or micrometers, you're gonna want to pipe that up and down a lot because like they're heavy, so they fall along.
These are really weird. If you just put the lid on, they pop out. You have to put that little pin. FYI. If you just put the lid on these, they don't close and they like shoot out. So it's the next one in the positive.
You can use these as tube holders. As little racks.
Oh, like yes.
No, if it's tea.
Oh, tea? Oh, I thought you said
Alright, this is 75.
It's still upstairs.
No, you're not in it.
okay.
Okay, imagine like number.
Go for it.
No, Qubit is still something you do yourself. Exactly. Once you're sure that, or there are plenty holders here, once you're sure that your DNA extractions have the proper amount, then you hand them over.
Okay, that's what I did.
Alright, guys. 400 and 500.
Oh, yes, you did. That,
Where's the tea?
with the NanoDrop. Okay, now your last step would be to measure with the Qubit. I think it's already in operation over there. And this is still, so when you clean up garage, how change? I guess that's the working together. Sorry, you
Okay, now we need to
it was relatively low with the magnetic beads. You have now all the fewer nucleic acids to differentiate the RNA. So it makes sense that
And we need this binding factor.
But you know, clean DNA, that is good. You just don't know what the proportion of RNA versus DNA is. So the Qubit will now tell you the exact DNA concentration. As soon as you're above, or as long as you're above 20 nanograms per microliter, you're good to go.
Running the finding number rate. Let's see that first. It'll be like,
It's up the north corner.
That's
No. It's like further in the portion. So I'm still about 2 hours in Kansas City.
Oh, that's nice. That's so funny because I'm in the complete opposite corner of you. I'm from the southwest.
What time are you from?
Exactly. I never heard of you.
Yeah. Most people haven't.
Is your population like under 1,000?
700. Yeah. Okay.
What the heck? I don't know. I guess it depends.
You mean the ones for the Qubit or the ones for the PCR? Yeah, because the Qubit, those have these special tubes. Again, they are sterilely sealed and they're sterile in there. So just
yeah,
when you put it out, like only 4 hours many as you need. And if I really enough, those are really all.
Okay, so that's all in your office.
And don't forget
Oh, just one
yeah, that's
Yes.
Yeah.
Did you already start a timer?
Okay. And then we need to incubate the mixture or teacher for
Just probably it's best to look at the protocol. First you do the status. And once it's correctly calibrated, then you can measure the status. So, part of these and mix the rest. If you need help right now, yeah, they are quite some. I know they don't really have that.
Too bad.
Do you want to wait here or do you want to just mix like shot it in the jar?
You have to keep it first. You have to. This is perfect. You're. I was just Dr. Louis said you like when you come again to get so you have.
Okay. And now we put on magnetic scan.
Standard 1, Qubit reagent at the bottom. So we need this image correct. Qubit reagent. Standard 2.
I don't know, but I really want to text Jamie today. Here's my 2 weeks.
Oh, those things can go too.
I'm really good at I tried to
If you want,
So this 100 is better than that.
this work like
And if
Renee
you want
used to
to
work
show
for
seconds and help you with it. And
Cascade. It's a CNA staffing agency. I mean, she worked at BIOL325L, but she literally just picked up, like, it wasn't like how we have weekends we have to work. It was literally, she just picked up whenever she wanted to. And made $21 an hour. Yeah, she also worked at the family med clinic, and it was not great. The providers, that was the main thing. Like, if she didn't set up the room, she literally had an NP come up and slap her on the back of the head.
It's like saying, you know, so like,
But that's just me.
Alright. Okay, here's a problem you already know the way out. No? Okay, because I was about to show the other group also here on the dock cam, so if you want, we can wait together. But I need about 2 minutes. If you...
Exactly.
Oh,
Because you're actually, it's not just online.
Fair enough, fair enough. So it's okay. I'll still show like that way you can, but you didn't use the same, the full sample, right? You only used half.
Yeah, so I'm like, how badly are you hurting for people? And you hired that.
Then let's do Qubit first.
and like,
No, it's not.
Yeah,
So-
like there are, I'm not saying that
Which
they're bad, but it's like,
is this is actually
oh, I need to research
I'm sorry, you shouldn't be able to go for 4 years
get DNA for
as, you know, like 20,
what you need to do is
maybe 24 years old and
take out let's say you have apples, then you have
take out the liquid and drive, but like,
then you have
PAs don't have to have an MP. Yeah, like, I get why they're doing it, but I feel like that could be big. I understand that. Like, there was a horror story of an Indian just prescribing medication for bipolar disorder for acne. Oh, and it literally, yeah, and she's been in therapy for like 15 months or something. It's like 15 months.
my mom tried and I failed it. So me and my roommate were saving up our cardboard boxes so when that comes out, so we can like shove all our cardboard boxes in like new ones or throw them around from who knows where. We're both like 3 hours away. So like you can sort of have
No, I would not take your car back.
but apparently the shelf is like, it comes with windowsill, so we have to move that 4 or 5 days. I don't have my roommate there, so I'm calling my friend and she feels so bad, I'm like, pretty much just
I would entertain that. I would take the risk. A lot of biochemists, like late biochemists, are biochemists too. Yeah.
where on campus do you live? I don't
fulfilled your 2A protocol. And this given you 2A. Then you go to 2B, meaning you have 5 of these other things. Then you have these 5 tubes here. Exactly. Then off that mixture, and I'll assume now this is your mixture, you should have 1 mole at a time. That you put into your negative, your positive, and your test tube. What was it? 195, right? Yes. 195, 195. And it's the standard 190, 190. And then, once that is done, you go ahead, standard 1. 10 of the standard 1, standard 1. Standard 1 is ready. Is it really? Standard 2. 10 of the standard 2 in here. Standard 2 is ready. 5 microliters of your sample to the sample. Sample is ready. 5 microliters. You shake each of these slightly, put it in the machine. Sorry. Standard 1. Standard 1.
Yeah, I'm used to it too, but like I know if like somebody that doesn't know what they're like, oh my god, it's like that.
And then
I remember
sample
like going
negative
to
control.
camp when I was younger, and I was like,
Does it make sense?
No, I went to a
Alright. You're also, if you like, we'll do it together.
I really like it. I was a doctor, and I was so nervous.
Giving the nerves,
I'm which I would want probably which probably does shed like fine like little stuff, but not like my labs that we have at home. We have 2 black labs that are both inside.
well, your sample definitely, the positive control, if you made it positive control, yes.
I want Yippee though.
If you took Reese's positive control, sure.
Yeah, I want to. I have a half to all that poodle, and he's the coolest dog. Really?
It's a learning experience, that's what you don't have to do it.
She stands at a window and watches these ones.
When that dog is 13, he's.
Alright.
I guess he's like, we think
By the way, you can come up, like you don't have to come and you'll see over there better.
Well, our TV does.
I can't tell that.
There's just like,
Well,
I guess.
oh, I see why. Look, there's the cable's up.
No, what's the magnetic?
It's called Land of Pause. It's just like a place for like kids dog that gets shots or gas. Sometimes they're fixed.
No, just wait. Is this already the mixture? Okay. But you only took half of it.
Yeah, no, my dad died at 13, so like.
My freshman year is like
Perfect. Yeah.
weeks, and
This gets into this. This gets
my dog
a bit. It doesn't get bad. Okay.
No, oh my god.
All right, everybody. If you want, I'll demonstrate. Thank you. Perfect. Excellent. I'll demonstrate the process of the. Or do you want to do it? No. You did it more recently than I did. But you know, you'll see better on the TV. It's wonderful that you look at me, but you'll see better at the TV. Well, you can watch one of the other. The other thing is I need to watch this TV a little bit as well. So, all right. This is the magnetic bead cleanup, which is one of the standard techniques in modern molecular biology labs. This is really something that, for example, for the cure will come up again and again and again. So better learn it now, and let me put on some gloves. This method is being used whenever you have some kind of contamination, or you suspect you have some kind of proteins in there or salts in there, and you just want to get a clean. Any enzyme that needs to be removed, the magnetic beads is your method of choice. What do you think, Lisa? Is this like how often do you have you used it? In the last year? It is very common. So it's a technique you're gonna learn. So what the group has done already is, let me see, is to put magnetic beads here into their sample. Right? This is their TS. This is probably their sample per se. And the reason why it's so darkish is that there are some magnetic beads already mixed into their sample. Like we can shake those a little bit and it'll become like literally grayish because the magnetic beads are mixed with their sample. The same with their positive control and the negative control. Now these beads, they sediment quickly. So literally you have to make them like an emulsion as much as possible. Just as an example, how quickly they will actually, can you see this? How quickly they will, I'll just put them over here, and you see this seems almost clean, this tube. Let me remove this again. Like, all of them will work the same way. Doesn't really matter. So you see how dark it is? I'll put them on here and 3, 2, 1. Swoop. It's clean because all the magnetic beads are held by the magnet. So the protocol is separated into literally 3 steps. Let me briefly look at it as well. The protocol is you add the magnetic beads, the DNA will bind to these magnetic beads. So until we change the pH, the magnetic beads will grab the DNA. So we will now put them on the magnetic rack, take the supernatant, throw away the supernatant, because the magnetic beads are holding the DNA. Then we're washing the sample, these magnetic beads, with ethanol, under which condition the DNA still stays with the magnetic beads. And then at the end, we will add some solution buffer. That's the only pH concentration until when the magnetic beads give away the DNA. And then we can no longer throw away the supernatant, but have to keep the supernatant. So in practice, that means this is incubated. Hey guys, just let me speak here for a little bit. This is incubated for more than 2 minutes. I know it is incubated for quite some time, but let's just assume it is incubated for more than 2 minutes. I'm now putting this on the stand. In fact, I'll do all 3. 1, 2, 3. Now they're already, if you look, they're already clean. The magnetic beads have already been pulled over to the side. So I don't really have to wait for 60 seconds. I'll take a little less. And I'm going to take off now the supernatant and throw it away. Can I have the waste speaker? And this is now one of the sensitive things. I'm going to open them one by one. And just take away the supernatant. See, I keep it here. The magnetic beads are pulled to the right hand side. I go in and just without disturbing the magnetic beads, taking the supernatant. See, this is just the supernatant. It doesn't contain any DNA. It contains proteins and secondary component, anything you want. Just not DNA. The DNA is stuck here with the magnetic beads. So Don't be shy. Many people are like, oh my gosh, I'm throwing away everything. No, 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 no. The DNA is with the magnetic beads. It's held by the magnets. Set it away. Now there's still a little bit left. Go in all the way, like every little drop of supernatant needs to go away. The stuff on the walls, that's your magnetic beads, this is where the DNA is. Oh, I see, no, don't worry about that. All right. So everybody was able to see that these tubes are now clean of supernatant. Now I'm going to add 100 milliliters of wash buffer. Did I have the wash buffer? Oh, that's right there. 100 milliliters of wash buffer. Don't add the elution buffer, the wash buffer. Because it's only the wash buffer is the one that will not interfere with the magnetic beads now. All right. By the way, you'll smell that the wash buffer is alcoholic, and that's good. That is what the wash buffer is supposed to be. Now I'm taking it off the stand, and the beads are supposed to be resuspended. Okay, the beads are still clinging to the DNA is still clinging to the beads is now being washed. I'll let you mix this a little further.
Thank you.
Okay, here's the next one. And here's the third one. So, this was the washing step. This should be equally, like it should be a nice emulsion of these magnetic beads. Then we put them back on the stands, one by one. And everybody please have a look at how quickly the magnetic beads are being pulled to the side. And you see the bead is, the tube is completely clean. Because all the magnetic beads are being, did you see that? And one last time. So the magnetic beads still contain the DNA. We have washed this with ethanol. It's again time to remove the supernatant. (100 microliters of supernatant are being removed. All right, and now lastly, we'll add the elution buffer. The elution buffer is now the one that with its pH value will have the magnetic beads release the DNA. So we're going to add, how much is the?
Who's that? Oh, it's back down.
Oh yeah, sorry, the protocol says to let it air dry for a little bit.
Yeah.
Come on, air dry, air dry, air dry.
Okay, it's fine. I was still in high school, so.
Okay, let's just assume it is properly air dried. And now I'm adding 20 microliters of emulsion butter.
Yeah, so this might be your first so sometimes 4. So yeah,
20 microliters of solution butter.
He's 3 years older. That's it? Yeah. That's alright.
20 microliters. 20 microliters. And 20 microliters again. I'll take them off now from the stands. And again, try to get a nice emulsion. So now the DNA disassociates from the beads under these pH conditions. We'll need to stand one last time in just a moment. But now it's important to really make sure this is nicely mixed. If necessary, you can briefly spin it down with the desktop centrifuge. Only spin it down slightly with the desktop centrifuge, just for a second, please. During this time, we have the incubation going on. There's only one last step left.
Yeah, I don't know.
In fact, could I ask, you got it down, right? Can I have 3 fresh tubes labeled with samples, positive, negative, control? Just 3 small fresh tubes.
Like, there's not a single thing by
I'll be with you shortly. I'll just finish it up. All right, perfect. Thank you so much. So your colleague is taking the three fresh. It's really important that you keep them properly mixed. They should not settle. Okay, perfect. Thank you so much. So, last step here. I'll put in the back of the stands, on the stand. And now the supernatant is the precious one. Because the supernatant contains the DNA, the magnetic beads
The standards are already
are no longer carrying the DNA. So don't throw away the supernatant now. So TS, I'll open your sample. So take the supernatant and only the supernatant and keep it. And make sure you don't have any of the magnetic beads in there. Can you see any magnetic beads? I cannot, which is good. It's just pure. This is now your clean DNA. Okay, clean DNA 1. Clean DNA. One second, yeah, I'll just finish this for everybody here. Because it did And lastly, the negative control. Now you don't really need to do the negative control. If you want, that's good if you want to practice, but it's not absolutely necessary. All right, and these are just the magnetic beads with dealing without any DNA. You can just throw those away, and the process is over. The 3 tubes that are that Kennedy now received. Can you briefly hold them into the camera, please? Those. They have each 20 microliters of that dilution buffer with your clean DNA. And you can go and measure those now, and you'll see that you have a much better 260 or 230 purity ratio. And that's it. You should measure them just that you see that, like how well it works. All right. Any questions regarding this? Magnetic bead cleanup.
We take the supermate and put it into
Good enough?
tubes and throw away those tubes. Magnetic tubes.
Ideally, both the NanoDrop and the just so that you see the difference. Yes, of course, that's good. We rarely use the old ones anymore, but yeah, in the left cabinet. All right. No, I think we only have 2 mechanics. No, actually, there's a third one upstairs. Do you want to go and grab it? It's that short one. Should be okay. All right. Yeah.
Sorry, guys.
The sample that you did last time is just a sample. Yeah, remember? There was only one that they pulled in the sample. But we discarded the positive and the negative one, right? So DNA being negative, usually DNA bring the sample. Well, that's okay. Because then
probably won't
the
be helpful, but
the other should not be affected.
where are you at?
No, let me try that because there seems to be some confusion. So this is now only, so what I'm drawing down is only Qubit maps.
Do you still have that marker? The what? The Sharpie.
So now we're this is now you had a sample cube, a positive control cube, and a negative control cube, right? Sample, positive control, negative control. And here you only rescued about, what was it, 10 microliters or so, give or take. Yeah? Whereas here you have like at least 20, if not more, in each case. So let's say you have 10, you have 20 microliters and 20 microliters. So in addition, you now have the following 5 tubes those are now the small Qubit cubes, you know? Gonna make them thus a little smaller, more delicate.
Please still make a difference. I think there's still some stuff from the last
Very underdressed.
on there.
And did you have it? Okay, perfect. This is standard 1. This is standard 2. They're set like they were extra, right? Each of these has already been filled with 190 microliters of that mixture of reagent buffer, plus 10 microliters of standard 1 or standard 2 respectively. Now here's your sample. Again, you have 195 microliters of your mixture of Qubit reagent plus buffer, and are weighting 5 microliters of this. This is your positive control, which again has 195 microliters of Qubit reagent plus buffer already in it, and this is weighting 5 microliters of the positive control. The same here.
Yeah.
So unless
I noticed when you I thought it was very light compared to other two. Yeah.
you no longer have 5 microliters, at least 5 microliters from earlier,
Because I'm doing multiple items. No.
you should be able to do the measurement.
Yeah.
Because all you have to give to the other group
So then
is 2 microliters. That's all they will need for the piece. So, literally with 10 microliters, and I think that's something you did say from last time. 10 microliters, you should have all it. You should have enough. It sounded like earlier as you were thinking using that sample for each of them. That's not correct, right? This sample only goes into this Qubit tube. Positive to the positive Qubit tube. Negative sample to the negative Qubit tube. Only 5 microliters each. And the standard 1 and the standard 2, they come in separate tubes anyway. Does that make sense?
Yes.
Okay. All right, that was Qubit.
Oh, we already got it.
This is the buffer.
All right. Here's this. Right? You need another chair? All right. You want to
Okay.
and what do you got? That's true. Because you already cleaned it, so you have more
Yeah.
with that positive control that you clean. It's not much, that's true. But it is clean DNA, so it's going to be sufficient.
Is it if I touch it? Yeah. Okay, thank you. Okay.
For your set? And you would theoretically not even need to clean it. Yeah, yeah. There will be some people who will not need to do that.
Yeah, we're not in 95. Can we get 1,000
All right,
microliters?
all right.
Do you get 190?
Which one were you on?
Sufficient plus 10 microliters of standard 1. That'll be a price. Then
May I make a recommendation? Can you
standard 2.
do the same? This one is like, oh, you just want to see what the standard speed
I can just take some sugar, 5 microliters of your sample. That's
You just take out the cube and you did the curve in there. And then start over again. You just click on the whole button at the very top. Right, exactly. Yes, has a mixed.
No.
Mixed standards. All right. Now, before you do this, slightly.
It's useless, right?
because the DNA is 6 to
Okay, I got these guys labeled.
Alright.
These fall. Sample.
Okay.
Because if it's not correct, it will be
Where do we find more of these tiny ones?
you see the other things, you would like
You wouldn't be able to, yeah.
Do we have to label 5 of them?
Are you even using these
But I don't label
So
them.
another calibration
Or
first,
someone else
correct?
does. So I label. So that's two standard ones, standard two, and new region. Okay.
So that's what you need to create your solution. So
Here's
you
how, like, for example,
Is it okay?
Yeah, go for it. It hasn't been mixed already, so you have to create your working solution. And I mix in 5 microliters of the orange reagent with 1000
I can get
microliters of the
Actually, we
so now become a working solution. So you can do that in a 2 something that will contain 1000 microliters. So once you have a working solution, so you mix
Is it set?
your heating component. So 190 microliters.
Oh, so we have to dilute. Oh, is that the math we were doing?
Plus 10 microliters of standard one.
Oh,
That
wow,
is
that
correct.
is
Let's
good.
say code.
You're right.
Then 190 microliters of working solution with 10 microliters of standard two. Okay, so you have standard one, standard two. So you mix 195 microliters, 195 and 190. Okay, you work your solution with 5 microliters of your standard. Okay, so you have, let's say standard one, standard two. The test sample and
In those cases, you would be
Yeah.
welcome to the
it's on the just let me know. Okay, sounds good. So you first have to do
Okay. What about the negative control? That should now show functions. Yeah, that's good. Because that tells you it's too low. So, you do suffer from a little bit of DNA shortage. It's gonna be okay. Just let the other group know that they will not be able to pick
each of them are going to have.
But see what DNA is, so you're ready.
This is standard one.
Next step is cost exchange, and then
Is it easier for me to put on here?
if you want, you can take the Qubit and bring it over to wherever you were sitting, but that way, it can keep working. You just have to upload it.
Thank you.
By the way, for those that haven't seen the Qubit yet, there is a cheat sheet here in the back, yeah?
Who's number 7?
Yeah, but you know, if you already have a group that is ready, you guys can share. Just let the other group know that they need to find somebody else. Alright, there's a cheat sheet in case you need it. But my protocol is better. So you prepare anything and then you back it
Okay, here's sample number 2.
Yes, no, it has not been mixed. This is why you have to, like the small orange tube is the reagent, the yellow big tube is the buffer. Sure, sorry, okay, so one, now, that by itself doesn't help. So you open it up, put it in kind of tube, then you have calibrated. Again, just gonna flip it a little bit because otherwise, like if you calibrate it wrong, then all the measurements later will be pretty confused. So literally, for the calibration. Alright, now you'll see a calibration curve. The calibration curve itself doesn't really help. You just know that you have a nice calibration curve, and now you start with your sample positive, so like control whatever you like. And now you can start really measuring the DNA. Yeah, so if you added 5 microliters of your blood into that. Perfect.
Oh, okay.
Alright, you have, now this is only good, right? This is positive control, 11.5 nanometers, right? So PCR, what's the positive control? It's
So this is every inline aid for. Okay, and it says H2 required.
Don't forget.
Oh, yeah, there's gonna be,
If necessary, like, you
standard
can like stick it out just like this, then we all come back. Alright, so
H2
for your
requires a
sample,
total of 200 microliters.
I know, so look, I mean,
I think you use 190 microliters of working solution plus 10 microliters of standard sample.
your colleague is.
Yes, the last step is PCR.
Okay, perfect. You know, this is why we have the camera. You can then later, I'll share the link tomorrow in class. You can like look at what we did.
Oh wait, is that the right?
Alright, this is the negative result.
No, that was
You see?
yeah, I just
Oh, sorry.
You're good, yeah, 'cause you're still liquid.
And remember, this is our DNA. No combination. Excellent. Whoever you're sharing
I'm glad I
with,
love that, because like, I know for a fact that we're still.
just to give you like an answer. These are great numbers. These are
I just think it's really bad.
Alright. Who needs the Qubit? Qubit is available. Yes. Well, hopefully.
Yes.
Yes. With sample, yes. Sample, so you see, in total we have. I'm gonna take that. So you have the full primers available. They're all listed here, right? Yep, and those your sister group, wherever they are. Yeah. Who would have been your sister group? Yeah, but who would have been your sister group?
Number 5.
So we were number 5. We created the 7.
Okay, then 7 is yours. Oh yeah, perfect. Give a second. If you're ready, that's it.
Have you done? You can
Sample, positive control, negative control. Alright. So, you have, animal, those are animal primers, fungal primers, plant primers, bacterial primers. For you right now you have 3 cubes in here. You have positive control, which you know are the sign of material.
Which high temperature bead do you use? The top one or the middle one? Yeah, that one.
4 PCRs with angle primers, another PCR with fungal primers, another PCR with what? 2 microliters samples, 2 microliters of the sample, 2 microliters sample. So
Yeah, that makes sense. Yeah.
to the PCR,
So do we need to take
you have 2 microliters of the sample. So the sample you need to have 3 microliters, which I think you have. If not, then just keep 1 microliter.
Yeah,
This one, you use the cyanotic hero primers.
Can you put the two of these on?
And for the negative control, you just choose any of these
No.
Okay.
and 1 primer here.
So we'll just do
I probably would take the bacteria,
control one, or sample one, sample two.
prefer
Qubit tubes, whatever that.
2 microliters. Yeah, actually, now that I think about it, yeah, I know I mentioned 5 earlier. I wasn't thinking of course negative control. You should be using 6 per second.
I think it might be 10.
Other questions?
Oh, 10. We did 10.
Well, we did.
Yeah, we did 5.
We'll just go with that. That makes sense. Yippee!
Each of these PCRs, before you actually start putting them together, you should think of, alright, what is the best needed temperature here, 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 and here, and here. Because we have 4 plates available. Each of these plates can have a separate enablement, but we have 5 different primer pairs. So one of them will share. It's gonna be this. But nonetheless, you have to figure out the optimality and
This is our negative control. So this is
That's for.
that makes sense.
I have SE1 and 2,
Oh.
but I don't know what that actually also
There. Yippee! So positive control is the only
Okay, so, okay,
Can you guys try out this? Is any of these I don't know what that's called. No, maybe it's the entire oh boy. So that's the case. Oh, there's some space over there now.
Yeah.
Yeah, you were right about the 5 microliters. Oh yeah? Yes, you are. That makes sense. Okay, because it didn't go up to 2. I'm sharing Okay, so we need, I don't know how many tubes.
So we have 4 different. Yeah.
So I just need to see like a simple number.
So these are the small ones. No?
Because he did say this was really
Everybody was.
he said I don't know.
Everyone's their
We see the calculation. Yes, it's my recommendation here. So for those who are not sure, like how many PCRs they should do, this is my recommendation. Your sample that you receive, you do a PCR with the animal primers, a PCR with the fungal primers, PCR with the plant primers, and a PCR with the bacteria-specific primers. Those are already 4 separate tubes. Then you have one PCR with the set of bacteria-specific primers, tube number 5. And then for the negative control, you can either choose any of these primers. I would recommend the bacteria, just because it's the most likely contamination you have. And then would be the 6th tube. Yeah, they're like 0.2, they each, somebody just, I think, look, they already there. Sorry, my apologies. Up here, because exactly. It's like the PA specific. Because then I'm kind of differentiate. The thing that they just said, yeah, the Qubit machine is ready for. Ready to go.
They're in that box over there. That's the end table.
Just close all of this for you. I'm gonna try my
I just think I'm going to do this year.
Primers, it's one, yes. These has to mix it up.
Are you using the pizza?
Because it comes in 2x
There's a
concentration.
I don't know where the other one went. I just did this in. Yeah.
in here, right?
Yeah, I guess I would take your time.
Concentration. And it needs to be as a one.
Okay, so I'm gonna go ahead and add 10 microliters.
Okay, so now it's everything.
And then we'll add 6 of water.
The old calculation, C1 then C1
And then we'll add everything else.
You have a 2x concentration to start with. You don't know how much you want to put in. You know is the 1x concentration at the end, and in total you have 20 microliters. Like that is a full tube.
Do you want to go
So you have to take 10 microliters of the PCR master mix. Of the water, exactly.
We'll do bacteria. So I'll just do it for 2.
Yeah, and again, every person has to do PCR, takes the caffeine set. And then, like, every table has one set. This was the
Okay, so we need to put 5 microliters in there, or 2, or ST1, ST2. And then that's what we do.
Yeah,
To measure DNA samples,
I think
slipped lens samples, input 5 micrograms. Insert 2 working specimens. In theory, it should be.
That is.
So, this tells you probably a couple of sorts.
for so many do,
If you look at the
so our
People move.
Okay, makes sense. So the ST1 are just
so take
to
You're the name. No, but that's the top. That is top reverse with one hand. So we'll get back to it, yeah. But before you go in, my recommendation is just what amount. Okay, so how much are you taking?
you take that in.
No, that is the two licenses. That is there is a SAT that was not the
I feel like the worst does all the fun.
exactly what he's doing. He started calculating the exact amount. It's if you go
Whatever is easier. Right now, I'm putting them on ice. I'll just have the water. Sorry.
that's very good.
this is something that you're going to is that when you know how much of the sample, I call it counting in the sample, also control the control. There would always be 2 microliters. You want to fill it in 20 microliters, so the question is how much of the primer, how much of the reverse primer, how much of the rest of it, and the water. Right? Now, it's not the same for all your
How do you know that?
but so then, figure it out. It's not too
Let's do the 5 first.
That's good. That is now, and now you measure the sample. It's going to be first. Very good. And you think of run sample. I will ask you how much of your initial DNA you put in the tube and then it's like, beautiful. You're really which is exactly, this is not pure DNA, right? It's exactly the amount that you exactly. Again, this is not a it's just a little bit on the lower side. We just call it controls. That's okay, but we can say, you just want to go with what you have. That's okay. Fair enough, and just think of it, it's just a little bit more positive control. Now that should of course be the lower. It's so small that it doesn't use it. So you're at the same time, here's the side. Those are great results. What? These things, you can throw them away. You have the measurements.
That's the solution.
this for a second.
Positive control is on the lower side. Otherwise, it's going to be
Oh, do you guys need water?
No, that's the magnetic thing.
This is the Qubit machine. This was $2,000.
So for us, you want to see.
If you don't need the magnetic stands, I'm happy to take them again because they interfere with your machine, so with your laptops and cell phones.
They are different, but it's just the price.
Oh, okay.
Not the volume of
Except for two already done. So the reverse and forward timers are okay. What about the PCR master?
so do 5.
Alright, that's 10.
It of course needs to be eventually in the 20 microliters. It needs to be a 1 and do you have something to try out? So you already know the initial concentration. You have 2x. But you know how much of the volume one put in? You do know you are 1x and the total amount of microliters. So V1 necessarily is
Just because of the
the 2x PCR mass mix you have to do 10 microliters.
on here. It might be 5.
And then we just add.
Oh. We already added. Oh.
So now you can already get 6 tubes ready. There are like 6 of these small tubes ready. Label them with sample A, like sample sub A, sample sub F, sample S, A, S, F, S, E, S, positive C, and negative.
So let's
And
do.
that way you know exactly what samples and what kind of size you want.
The positive we should have that. 8, 5.
So then after we add the practice, then the
So,
Alright.
no, that's the same.
If you want, you can, like once they're done, you can just bring it over. You don't have to literally, but yes, it's here. The question?
Yes, so we're trying to
It's
figure
kind of
out the primer pairs, but it's tell which is which. So we
Yeah, it's. Do you mind if I use this for a second?
Oh yeah, you have to use this.
Okay, no problem, no problem. If you. So you have the animal-specific ones are LCO and HCO. These always took like the first few letters. The final ones are ITS4, ITS5. Plant ones, TRNH, PSPH. And bacteria are CAP5 and CAP8. And then the cyanobacteria ones are cyan1 and cyan3.
Okay, thank you. That helps a lot.
Yeah, it's like those are the official primer names, and we have to somehow squeeze it on there.
Can I get primers?
Is it fair?
This was our first DNA.
Well, it all depends on the initial instrument there. It all depends on the initial calibration. You know, the calibration was off, it depends all of the measurements were off.
you still have everything there.
Yeah.
Or did you throw the Qubit?
We still have it.
Well, you can just remeasure them. If everything is there, no, you can there. Okay.
This is our SEC one. Yeah. It's a
Wait a
How
minute.
much are each parameter?
Maybe it wasn't calibrated right.
Okay.
That's what's good.
Yeah, that's what I
Yes. So you need the machine again, and I know this is like the first using the Qubit the first time around is a bit confusing. You'll see it's the easiest machine ever. Just the first time around there's a little bit of a learning. Let's go back and let's gradually get it. Are you using the Qubit? Qubit is free. Should we just take it over?
Then under 95.
So if you start over, the first one. That's all right.
So what am I doing?
Alright, so
Oh, am I doing it? Oh. So I guess
but make sure that
and does it say no, just one microphone.
necessary. Put it in there, and then click on read stamp. Then we now do the second sentence, make calibration. You see? Now it's correctly calibrated, and now you can put in your direction. That's like your sample. Or like on the control if you want to choose first.
Oh, I'm sorry.
But okay, you can do PCR with it. It's not going to be but those are your own ones. There is nothing easy. Yeah, that means you don't have let's try the negative control. This is the result.
Alpha fumble, right?
Write down correctly.
What?
So that if you got like there's right now what you're selecting, our recommendation is
Well, the PCR also.
Doesn't make sense. Yeah, if you want, you can take it and bring it to because otherwise it might be a little bit too further than like here. Yes, you hand over the sample. Now you already calculated this, that's great. That's perfect. And don't forget, before you get ready with the PCRs, you want to calculate the PCR unit temperature. Explain it all here. Otherwise, you guys always just follow the recipe. Yes, absolutely. Or alternatively, over there, close to the camera, and people see what you're doing.
And you see how
Smile for the camera.
Okay.
All right. You're
That's all I.
good that you're doing the PCRs, but you know, the PCR machines are not ready yet. They need to be programmed first.
We already did this. That's preparation of it.
Okay. So turn
This
it
one
on.
there.
The numbers are
Yes.
good,
And then put the little
but you still have to program the machine.
read standard?
Insert it and then
You gotta
the instructions are so long.
Read standard?
Yeah.
You need a. Can you give them?
Find us somewhere.
I want to go to a TO school though.
Yes.
So my.
All right. Here's a little reminder, everybody, because I see many people already getting ready for the PCRs. We haven't programmed any of these PCR machines yet. You will need to know what the optimal elongation time and annealing temperature is. And this needs to be calculated for each of the primer pairs separately. So you have a total of 5 calculations. Now that doesn't mean that every one of you has to do these 5 calculations. I explain how to do them in the protocol. But maybe we can agree on who is doing which one. Who wants to do the PCR protocol for the animal primers? Animal primers. Who wants to do the PCR for the animal primers? We need a volunteer going for. Huh? Sorry? Zap? Okay, good. 3, 2, 1. Their group is doing the animal primers. Who wants to do fungal primers? We got fungal primers for offer. Fungal primers. Plant primers for offer. Plant primers. Bacterial primers. You were just agreeing to something? Bacterial primers? Bacterial, okay, too? Okay, bacterial primers. And lastly, cyanobacterial primers. Cyanobacterial primers. So in each case, you figure out the annealing temperature and the elongation time. And then your representatives come please over there. I'll wait at the PCR machines and we program them in.
So this is to measure DNA samples. So you did this run samples of the STD.
Yep, so we did standard 1, standard 2.
Right.
Have you read the standards for that?
If you need
Read
more,
standards.
Oh my god.
We did read, I clicked read standard after standard 1, then put this one in and then
That's this.
okay, so just take that.
And then
Okay.
And this is your class
measure
packet?
DNA samples like the run samples.
Oh, brother.
And then it says input DNA volume 5 milliliters.
But you
I think it's insert to or more concentrate.
Oh boy. So much
Repeat procedure. I.e. your DNA extraction positive and negative.
I still can't tell if I'm based on the sounds.
I know what DNA down to
Oh,
Okay, all of us are
Like your numbers.
so now,
Wait, that's your negative. That's right. I would ask them. I'd ask them just to be safe.
both of those
any of this crap anymore. You don't even.
One second. Guys, I have one more thing for you. I just noticed something here. Many of you are starting the PCR, like getting the PCRs ready, and understandably, write down on the tubes A for fungal, A for animal, F for fungal, P for plant, and B for bacteria. Just be aware, the others use the same letters. So everybody's going to share the same of these plates. So we have lots of A tubes in there for animal, and you might not know which one is yours. So if possible, make some marking on the side of the tubes to indicate that this is your sample. Like for example, Kennedy, you can do like a K, like a circle or something around it. That way you know, because you have an animal, a fungal, a plant, a bacterial, and a cyanobacterial tube, and probably even a sixth tube. And if all of them have a small K on the side with a circle, you know this is yours. And I'll take yours. You can do like, I don't know, like a little star or something on each of the sides. That way, that's your identification. Just be aware. Otherwise, next time, later today, I'll just take them out and put them in a rack. And then you have to figure out next time who's whose. And this might be quite difficult if you haven't marked them. Well, initially the idea was like if your sample, if you're providing a read, you exchange it with number 4, but I think maybe I've already exchanged it, just find the next best group. We have an even number. So eventually every group will get up here.
So this is the negative.
Negative control is not right. Can we calibrate it again? This is the most important thing is the calibration. You just there. Can you put it in sample number 1? Sorry, standard number 1. Never forget, always flip. Qubit is very sensitive. As soon as the thing settles, then you get, especially with the standards, read standards. That's not run samples, it's read standards. Okay, you insert standard 1, read standard. Okay, this is number 1. This will not really give you an interesting number. You don't even need to take a picture of it. This is irrelevant. Yeah, this one is irrelevant because it you can calibrate it again. So now you added standard number 2. And then when you see you get a calibration curve.
You don't like talking about it, or are you shy?
All right, click on read standard again. All right, now all you know, it has been correctly calibrated. Like the numbers here of this standard doesn't really matter. So now you open it up again and click on and like put in your sample tube or your negative control, positive control.
You're so good that you're on carbon.
And now you click on run
So
sample.
like that
This will not be the real.
makes you look
You
strong.
put in 5 microliters. I set up the protocol so that you can always just click on the default. But that is your real, like that is, for a PCR, it's a good number.
Okay.
14.2 nanograms per microliter. Okay. Now let's put in the negative control after. So it's a picture.
Wait, does it matter?
Oh
No, it doesn't matter which order you take them. That was the negative control or the positive? Negative. Good. It's almost 0. Now let's see the positive control, which should be higher than that, obviously.
So you were labored.
Good. You did it right. You did it right.
You did good.
Yeah. Wait, could you take a picture of this?
Yeah.
That is your, these are your twin PCR, like, how you find the PCR together, that's good, that's all you
Yeah.
Where they did.
This is something you don't need to do.
Yeah.
This is the PCR protocol where there are only 2 numbers that are need to be figured out. And here, specific for each primary pair. The animal specific primary pair has here 1 unique number and here 1 unique number. And the funnel has two
I need to look at it. 82 was
for this, you can use this layer.
Is she like a guy
And for
or
the
what?
time, exactly, you just copy and paste the entire sequence in here. If you have a picture of 3, and then you get the right number. And this one, you check on page 3 what the average like this.
I guess he said he doesn't have
By 1,000 base pairs, that we know. You're looking for the seconds. Right?
Yes, sir. Yeah, that's good. Well, it's like 11.3 nanograms per microliter. It's not zero, right? It's a reasonable amount. So the threshold typically is, if you want to guarantee that the PCR works, you want to have 20 nanograms per microliter. Yes, it's true. You're a little bit below that, but the PCR will work, even though that is slightly lower concentration.
Okay.
Yeah, it's not a problem. 20 nanograms per microliter is kind of like, it's almost a foolproof number. You're a little bit under that foolproof number, but it'll still work. So you can exchange and tell the other one, yeah, do the PCR as a great. Correct, yes. Yes, if you do the blind specific one,
Yes.
primary sequence there.
Okay.
So you see in the neos at 63 degrees Celsius. Now, typically, so as I write in program, if you want to be on the safe side, you go below. Now, what exactly? Like how much? Between 3 and 5. Between 3 and 5 degrees centigrade, you go below that official number, just to be on the safe side. You see, because at 63, these primers start to bind. 60 is the effect. Yeah. So in the protocol, you're talking about 60 degrees Celsius. Yeah. In the PDF.
They never
No. 60 would be optimal, and then you put in parentheses 60. There, here, 60, and then we parentheses 63, because we want to share with some other group. If somebody has 61, good, then we could share with them, right? If somebody has 55, we don't want to share with them, right? And the second thing is the extension time. You look at how long the fragment will be. Again, in the PDF page 3, it tells you how long the average fragment is, and you know that per minute the back polymerase can do 1,000 base pairs. Well, if 60 seconds are 1,000 base pairs, then how many seconds are let's do it at the maximum 500. So if 1,000 base pairs require 60 seconds, then 500 base pairs require 30 seconds. That's exactly.
So, the whole thing about the analyzing
Well, let's get over there.
No, well, that's general. If you want, so, what are the numbers that you're grabbing?
Wow. Can I see slide that?
So my sample range was 2 over negative.
Okay, no. For those of you who are wondering how to do the calculations for the PCR protocol, for the PCR protocol, I'll explain to you one more time. Yeah, in case you're wondering, oh, how do I need to do the calculation with annealing time and annealing temperature and extension time? Then I'll do it now. All right. The annealing temperature depends on the exact parameters. So use like the people in the picture were.
Did you like
calibrate it. Did you like when you did like the first two, like the first one too? Did you like the calibrate, not like don't read it as a sample?
Yeah, so like click the back arrow and then do that.
That's crazy. Yeah, we didn't like that.
I didn't even see this, this you open up on first page, and it gives you calculus. You place the primary sequence of all the cells up there, and it will tell you the exact temperature at which the small fragments you start to anneal and you start to hybridize with the
I'm not gonna understand it.
Yeah, she's doing that.
Or she's going back.
Yeah.
we'll do it
There's
together.
not
Let's
done
stop
with
this, and then we'll start programming with the okay, so you see 60 degrees Celsius on top, right? Is the raw temperature that's 8 centimeters below 87. So for the actual time, right, that's 7 degrees Celsius. For you, it'll be slightly different. Let's do the second number, which is 1. So that was the temperature. The second thing is the extension time. So if you go back to the drill, the final law. So let's take the 1,500. Let's go further back down to and you see, approximately like the taxon race needs for every 1,000 base pairs, it needs 60 seconds. So for 1,500 base pairs, it would need a minute and a half. Now you have the protocol speed up there, so we could start programming and you can do it right. All right.
Did we let it down? I think so. That's what you were doing. Oh, you did it.
Whoever is ready for the first programming, we program this sounds worse than this.
This is the
That's true. If you haven't switched yet, it's
That is like, I think
Let's
about
start first thing in the morning. But yes.
this is like our test.
so T for secret.
I never able to change. Just like this. You can start this. Yeah. So, why don't you
Wait.
Yes. You both click on new protocol. Then the default protocol will load. Let's first add the name. F25 for default 25.
No.
Underscore or dash.
These are really nice.
Which you would find. And now, let's also do. Let's see, your
We did that part. Okay, we're doing feather.
Oh, feather was the T part of it. Just gonna find
and then which one did you choose? Flat.
Okay.
You use arrow, maybe for flat, or maybe flat. That's a little.
For a sample, we got that.
Okay, and click OK. That way,
I was just making sure.
95 degrees. I know. That's fine. So,
Negative, I think it's
exactly. So,
Did you do that again?
Okay.
You just want to restart?
Actually,
you have 4 shifts.
No, high sensitivity, right? What?
This is my job. Okay, turn it that way. I don't know what he's not really just turn it out.
What makes a difference?
Frankly, though. So now it's like
I'm really trying to.
The 30 volts with the initial one being 35 volts. And then you just plug them in.
which one of these are the forward, reverse, and the master, or the PCR?
And again, you're ready to start.
So it depends on what you're trying to do. What are you
The cyanobacteria.
So
And what we do right now,
But there's still really basic stuff like I don't like wedges or like dashes. Like I still don't get that 100%.
So these are the parts.
Literally did everything right.
So if we have this.
right here. So plants, we have animal, fungal,
Okay.
plants,
Okay. I'm with you.
and cyanobacteria.
Okay. Okay. I'm with you.
Bro.
It needs to be cyan. 108. I've seen that. And we use that for the
Right.
Well, there's 108 and then words of music.
Yeah, what about you? What's your
We use both. These ones are forward and one's reverse. Oh my gosh.
I need to practice.
I would recommend if they have time to do a PCR true for negative.
I can do the highest.
But it's not, like that is the least important thing of this.
To the babies.
If you have time, sure.
Which one is this?
I wasn't positive.
Oh, you was not supposed to. You asked the kid, but I think what he says. You want to know? I think we're gonna leave.
Please.
All that time times 3 or whatever. And then like
You're probably
that's
right
just
about
but like I don't like seeing how I'm like really competitive. And I don't really stuff in time. I put my calculator so I can see that until that 1 hour.
We do 6 of the water.
He says, yeah, I'm taking it down.
Oh, maybe that's
That's
Yeah, well done. Start. Anything we had
Directly one. No, that's what it is. Yeah, 11:30. So like, at least yes, time want to go and stay for the
Alright, what you got here?
So our sample and negative are arranged, and the positive is extremely low.
Can I see you calibrate first?
It's supposed to be
Don't forget to fill it. Exactly. It's okay. Maybe like what were you? What DNA were you extracting?
It was from the 4, but we already traded.
It's like the one. Yeah. Probably there was not too much DNA in there. They could have been.
From the NanoDrop reading, we had 4 NanoDrop.
Yeah, that was very low. But remember, okay, now it's correctly calibrated. Fair enough. So give me the sample. Yeah, okay. You correctly calibrated it, yes, you don't have enough. Under those conditions, I would recommend you take one of these as positive controls and treat it as if it's your sample, because the other people you're exchanging this with will at the very least see a positive band for bacteria. You don't have to do it this week anymore, because we're basically out of time. You can start the PCR early next time. Yes, barely, but yes. It will work just in the nick of time next week. If you like start first thing next week. I mean, if you want, you can stay out there around for 10 more minutes cleaning everything up. But
We're just waiting until cyanobacteria and
use that as a sample. Exactly. Pretend that is your sample.
I got a question.
Yes. Before
Who has not swapped? Like stuff. Okay, so we're good. There's a group that is missing a partner.
Really? Well, you guys haven't swapped either, right?
We can swap it, yeah.
Okay.
I don't think we swapped it.
You haven't swapped either?
Well, my part is somebody like group 1, because I was 12. Group 12. But
We have a match. We just have a match.
but like my partner's not here. My original partner's not here.
I'm sorry, dude. No, let's just make this a match.
Okay.
All right. Is it about the PCR or is it something else?
I just wanted to know how to calculate the time, just theoretically.
Okay, no problem. Good.
Okay, so here's the calculator, whatever.
Yeah, but that is for the annealing temperature. That's good. You copy and pasted it in. Okay, perfect. You see, 48 degrees. Then you don't need to change. None of them. Oh, that's true. Fair enough. You change it. That's totally okay. You were actually right to change it. I just realized that the others should have changed it.
Okay, no, I just wanted to know like how if I did it right.
You know what? It's actually, it's a good point. No, you actually did more than right. I just realized something. What was it? For those that already programmed it, when didn't remind you of it, did you select tag polymerase as the because then your annealing temperatures might be off. Please check this one more time. You can easily modify this. Just when you select the temperatures, no. I mean on the calculator, on this online calculator, the annealing temperature. When you copy and paste it, the DNA sequences, it should say tag polymerase as a choice. Right? So, since we're basically out of time, we're 1 minute above official time. Whoever wants to start the PCRs now should do it. Other than that, if you have the PCRs prepared, if you have the PCRs prepared to start it, if you have not prepared the PCRs, then please do it next time. And then bring all
the ingredients back, they need to stay on ice. Below that.
How do I choose that?
Just keep it as well. For now, no proof that you're not going to use them probably anymore, but just keep them.
Zach, did you get the cyanobacteria?
If all you need is have it placed in there, I'll take care of these applications. Put it in there.
Where do we store
Just make sure we know where your samples are on the base. Okay, then let's just put them. This is sample fungal.
Hey, Hunter. Is this ours does the same thing. So what is that this negative control? No, it's our same. It's the same thing. It's the DS. Did you do your other two? Yes, check your positive. See if that does anything. Make sure you plug it.
This one. The 43.
She said for some reason.
The sign of a cure.
I would retry your sample because maybe that left
You can 45 years. Okay, then this is the 5 ones.
Yeah, we're good.
What did you say for you?
Well, the samples, everything goes into the blue box because then will be frozen. Just make sure. Oh yeah, so the other sorry, no problem. That also goes back. And just make sure that the sample related is somehow so that you recognize it's yours. Yep.
Were those just over there? I'm assuming they don't need it anymore. Do you guys need this anymore?
No.
That's a good question. Dr. Qubit.
So perfect. Bacteria.
What do you mean? They're calculating the standards that or whatever. How much you want to
I think if he doesn't, I'm going to worry about it.
Whoever wants to start the PCR, give me your samples now, then we'll start.
Yes.
The 0.2 milliliter jars should be behind here. The 0.2 milliliter jars. Who has a 0.2 milliliter jar too? Oh, it's right there. Thank you, much appreciated.
I'm sorry, Zach. You ready for OChem exam tomorrow?
We're getting ready for it.
You're getting ready?
Getting ready.
I think I'm just gonna like lock in super hard today.
Is this yours?
And I do wonder how is it possible that we have so many more bacteria samples and the sign of bacteria, so there would be like so 4 would be 8. Yes, and of course, if we have the we have the negative samples as well, so it would be yes, that's perfect, exactly. That's exactly. Okay, yours is the better, then no rush.
Are you ready for OChem exam tomorrow?
Hopefully. I don't know, I'm coming at the point where I'm like, why do I even study anymore?
I don't know. Like I'm at the point where I'm still brand new studying, so like just finding not even. I'm starting to understand acids and bases.